I am Dr. Khokon Kumar Dotto. My Christian name is Dr. David Moon. I'm working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is MicroRNA-34a and Cancer Immunotherapy. Immunotherapy is defined as the application of any component or ability of the immune system in the fight against any disease. Immunotherapy is not limited to cancer only. Scientists are trying for a long time to use the power of the immune system to tackle this deadly disease. They are trying to get benefits from different angles and targeting different components of the immune system in the human body. The major target of different approaches for cancer immunotherapy includes number 1, dendritic cells, number 2, T cells, number 3, NK cells, number 4, antibody, number 5, cytokines, number 6, interferon, number 7, complements, and others. Dendritic cells works as antigen-presenting cells in the human body. They present the antigens to the tumor and cancer. They present the antigens of the tumor and cancer cells to lymphocytes and activates them to kill the tumor and cancer cells. In cancer immunotherapy, dendritic cells are activated using different approaches like vaccination with autologous tumor lysis or using peptides from tumor and cancer cells. Antigenic tumor peptides are often used in association with different adjuvants like granulocyte macrophage cone-stimulating factor, GM-CSF. Dendritic cells can be activated using antibodies against the surface molecules of the dendritic cells. Dendritic cells receptor like TLR3, TLR7, TLR8, and CD40 are usually used as the target for the antibody for stimulation and activation. Dendritic cells are even stimulated and activated outside the body and then injected back to the patient's body. The other approach for cancer immunotherapy is the engineering or modification of the T cell receptors for the enhancement of their efficacy and efficiency for the recognition and the elimination of the tumor and cancer cells. The T cells with chimeric antigen receptor is called CAR T cells. Fighting immune cells like T cells, macrophages, neutrophils, NK cells, and other can also be activated using antibodies raised against the surface molecule of them. PD-1 and PD-L1 inhibitors are also well known as cancer immunotherapy. Both PD-1 and PD-L1 inhibitors are antibody. PD-1 expressed by T cells and inhibitor antibody used to activate T cells to kill the tumor and cancer cells. Antibodies are often used in the conjugated forms where a toxin or radioactive part are attached to kill the tumor and cancer cells. Antibodies are usually made humanized to escape the unexpected immune reactions in the cancer patients. Complements are also used in the immunotherapy. Complements can help to kill the tumor and cancer cells after the binding of the antibody with them. Cytokines also help to mount immune responses against tumor and cancer also. Interferons and interleukins can be mentioned here. For example, interferon alpha and interleukin 2. In 2018, Nobel Prize in Physiology and Medicine was awarded jointly to James P. Allison and Tasuku Honjo for their landmark discovery in the cancer immunotherapy. Professor Dr. Tasuku Honjo and his group discovered programmed cell death protein 1 or PD-1. PD-1 belongs to immunoglobulin superfamily and expressed on T cells and pro-B cells. PD-1 can interact with two different ligands, PD-L1 and PD-L2. They found that PD-1 works as the immune checkpoint break. When PD-1 is inhibited with the immunotherapeutic drugs like antibodies, the immune systems of the human body attack the tumor and cancer cells. PD-1 inhibitor immunotherapeutics seems to be effective against a large number of cancer types. MicroRNA-34a is a well-known tumor suppressor. Now it is known that MicroRNA-34a directly regulates the expression of PD-L1 in cancer cells. G. Wang et al. Published a research article in March 2015 in the Journal of Cellular Signaling entitled Tumor Suppressor MicroRNA-34a targets PD-L1 and functions as a potential immunotherapeutic target in acute myeloid leukemia. They found that MicroRNA-34a directly binds with PD-L1 3' untranslated region UTR and regulates its expression. Targeting MicroRNA-34a in the cancer immunotherapy should work nicely for the cancers that express PD-L1. The use of MicroRNA-34a for immunotherapeutics has already been filed for patent in the USA on 31st March 2016 by Mina Therapeutics International. P53 is one of the most known tumor suppressors for human cancers. Almost 50% of all human cancers carry some abnormalities with P53. It has been reported that P53 directly regulates the expression of MicroRNA-34a. Nina Ravera Sapira et al. Published a research article in June 2007 in the Journal of Molecular Cell entitled Transcriptional Activation of MicroRNA-34a Contributes to P53-mediated apoptosis. Francisco Navarro and Jerry Lieberman published a research article in July 2015 in the Journal of PLOS ONE entitled MicroRNA-34 and P53 New Insights into a Complex Functional Relationship. They found that MicroRNA-34 also regulates the expression of P53. The trans-activation inhibitors like TP53 and MDM4 are directly regulated by MicroRNA-34. MicroRNA-34 regulates at least 4 genes that inhibit P53 in the post-translational level. MicroRNA-34 overexpression increases P53 protein level and the stability of P53. About a quarter of the messenger RNAs of p53 network are regulated by MicroRNA-34. A fifth of messenger RNA's target of MicroRNA-34a also bind with MicroRNA-34b and MicroRNA-34c. Khokon Kumar Dutta et al. Published a research article in September 2007 in the Journal of Cancer Science entitled Association of MicroRNA-34a overexpression with proliferation is cell type dependent. We checked the expression of MicroRNA-34a in 177 samples of human cancers of different types and found high level of expression of MicroRNA-34a in 50% of the samples. The findings support the relation between p53 and MicroRNA-34a with the evidences of diminished functionality of p53 in 50% of cases of human cancers. MicroRNA-34a also works in p53 independent ways. Abnormalities with MicroRNA-34a expression are found in almost 100% cases of human cancers. But the differences in the expression suggest that MicroRNA-34 may contribute in cancers using different mechanisms and pathways. The effector mechanisms of MicroRNA-34a may differ in cancers even originated from the same cell type in the human body. We also for the first time succeeded to localize the expression of MicroRNA-34a in lymphocytes in the germinal center of spleen using in situ detection approach. It would be nice to mention here that the functionality of different genes may differ in different developmental stages of a single cell or single immune cell. Martin Hurt et al. Published a research article in January 2019 in the Journal of Cell Death and Disease entitled MicroRNA-34a, a new player in the regulation of T cell function by modulation of NF-kappa B signaling. MicroRNA-34a regulates at least 14 genes of NF-kappa B signalosome, PLCG1, CD3E, PIK3CB, TAP2, and NF-kappa B1A are included among target genes of MicroRNA-34a. Activation of CD4 positive and CD8 positive T cells entrains a gradual increase of MicroRNA-34a. Martin Hurt et al. Published a research article in July 2019 in the Journal of Immunotherapy of Cancer with the title MicroRNA-34a as a hub of T cell regulation network. The group recommended MicroRNA-34a as target of intervention towards a modulation of immune responsiveness of T cells in broad tumor context. Meng Tao Zhou et al. Published a research article in September 2018 in the Journal of Carcinogenesis with the title MicroRNA-34a promotes MICB expression in hepatocytes. They reported that MicroRNA-34a regulates the expression of major histocompatibility complex class 1 related sequence B, MICB protein, a ligand of NKG2D receptor. MicroRNA-34a can both induce and reduce MICB expression by upregulating ataxia telangiectasia, and RAT3 related ATR protein kinase, and downregulating the transcription factor E2, F1 respectively. It is to mention here that Khokon Kumar Dutta et al. In 2007 reported transcription factor E2F3 as a target for MicroRNA-34a. We also discussed the possibility of interaction between MicroRNA-34a and E2F1 to 4 in different cell types. Rong Chen et al. Published an article in January 2016 in the Journal of American Journal of Cancer Research with the title HPV E6 P53 mediated downregulation of MicroRNA-34a inhibits Warburg effect through targeting LDHA in cervical cancer. They did a gain of function study with 12 microRNAs that are frequently found with downregulated expression in cervical cancers. MicroRNA-34a was one of them. MicroRNA-34a directly suppresses lactate dehydrogenase A, LDHA, and inhibits Warburg effect in cervical cancers. Lactate dehydrogenase A, LDHA, catalyzes the last step of the glycolytic process. In the study, they used two different cell lines, HeLa and SHeHa. They found that knockdown of oncoprotein E6 expression of human papillomavirus in SHeHa and HeLa cells by siRNAs lead to an increased protein level of P53, decreased level of MicroRNA-34a, as well as reduced Warburg effect. Previously, lactate was known only as the metabolic end product, but increasingly it is evident now that lactate is related with diverse type of functions in tumors and in cancers and in the regulations of different pathophysiological aspects in the body of a cancer patient. Lactate is now known to be involved in the epigenetic regulation of gene expression. Lactylation of histone proteins in the lysine residues plays important roles in the regulation of gene expression, including the oncogenes. Lactate was once believed as the metabolic waste, but it is now known that lactate is used as an important fuel for energy production and even can be used by the Krebs cycle. Lactate plays important roles in almost all the states of carcinogenesis, from tumor formation to metastasis. Lactate plays important roles in the tumor and cancer microenvironments. Lactate is now known to play important roles in the modulation of immunity. Immune cells infiltration is almost always found within the tumors and cancers, but lactate suppresses the immunity inside the tumors and cancers. Lactate helps the immune evasion process for cancer cells. Lactate acts as negative regulator for both innate and adaptive immunity. Lactic acid affects the intracellular and extracellular pH, which is an important regulator of cancerous growth. Lactate is also known to be involved with other functions too. Lactate plays important role in the angiogenesis, wound healing, and in other regenerative processes, memory formation, and others. Rapid growth and proliferation is characteristics of early embryonic development. The presence of high lactate is found in the early embryos. High lactate also works as important factor for the stem cells, which show a similar growth pattern like cancer cells or cancer stem cells. Jennifer Ding et al. Published an article in 2017 in the Journal of Cancer Biomarkers with the title Elevated lactate dehydrogenase LDAs can be a marker of immune suppression in cancer. Interplay between hematologic and solid neoplastic clones and their microenvironment. Alexander Margaret et al. Published a review article in March 2018 in the Journal of Frontiers in Oncology with the title Metabolic Symbiosis and Immunomodulation. How tumor cell-derived lactate may disturb innate and adaptive immune responses. Nuno Santos et al. Published a review article in October 2019 in the Journal of Immunometabolism with the title Lactate as a Regulator of Cancer Inflammation and Immunity. It seems that MicroRNA-34a targeted cancer immunotherapy will also help to activate the immune responses by inhibiting high lactate production by cancer cells. That means by inhibiting the overboard effect in cancers. Finally, I would like to wish happy, prosperous, and dignified life for you, everybody. Thank you.